Hello everyone, my name is Pinaprapha Tangpradapkiat. I'm here to present to you the project called Effects of Gardenia jasminoides fruit extract on attenuation of paracetamol overdose-induced hepatitis in mice. My advisors are Professor Domporn Weerawatakanon and Lecturer Mani Ratchayanupatkul. So firstly, let's begin on the background and literature review part. The dire need for this research to happen is demonstrated through the prevalence of paracetamol overdose-induced hepatotoxicity. As paracetamol is one of the most common analgesics, along with its increased prevalence with other combined tablets, for example, pain relievers and antihistamines, paracetamol toxicity could be witnessed worldwide. Whether it contributes to 56,000 emergency department visits and 500 deaths each year in the US, and it is also the leading cause of liver transplantation worldwide too. Next is the liver, which is the target organ of interest in this study. Liver is the largest organ in the body. Its subunit could be classified into two main ways, whether it's microscopic unit called hepatic lobule, consisting of central vein, portal triad, and hepatic sinusoids, or functional unit called hepatic acinus, or 3D aggregation of the hepatocytes. Liver could be divided into three zones of oxygenation. The zone 1, which is periportal zone, it has highest oxygenation. Zone 2 is intermediate zone, lower oxygen than zone 1. And zone 3 is called perivenous or centrilobular zone. This zone has the lowest oxygenation along with highest biotransformation activities, which is the zone that is most involved in paracetamol toxicity, in a sense that it would be the most vulnerable one. The next term is hepatitis. Hepatitis means inflammation of the liver. So the cut point for acute and chronic one is 6 months. The cause of hepatitis could be many things, for example, infections, toxic damage, which includes paracetamol overdose, or immunologic damage. The slides explain the paracetamol overdose toxicity. The minimum toxic dose in adult is 7.5 to 10 grams, whereas in children it is more than 150 milligrams per kilograms. Acetaminophen or paracetamol 90% would be conjugated into sulfate and glucuronide, which gives off a non-toxic metabolite. The remaining 10% would therefore be metabolized by CYP2E1 into a NAPK. NAPK is a toxic metabolite. However, with the presence of glutathione, this will further conjugate the NAPK into the non-toxic metabolites. However, in a state that acetaminophen or paracetamol is excess, the conjugation to sulfate and glucuronide pathway would be exhausted. Therefore, the remaining acetaminophen would be metabolized more through the CYP2E1 pathway. Would this happen until the relative glutathione depletion occur? The excess NAPK would form a protein adduction within the hepatocytes. This will at the end lead to hepatotoxicity. Gardenia jasminoides is a shrub found in tropical and subtropical regions, including Thailand. Its fruit, shown on the left picture, can be harvested and turned into herbs and industrial purposes. The fruit contains many phytochemistries. The most abundant one would be the iridoid glycosides and flavonoids. Among all iridoid glycosides, the most common one is called geniposide, whereas the second most common one is called genipin. Beta-D-glycosidases can hydrolyze geniposide into genipin. Geniposide is considered a more stable form than genipin. Industrial use for this herb, it could be used as natural colorant, additives in food, and artificial dye. In terms of pharmacological use of Gardenia jasminoides, it is found that the fruit extract could be used as anti-inflammatory, anti-allergic, anti-tumor, antidepressant, has beneficial effect on cardiovascular system, nervous system, digestive system, for example, protecting against acute pancreatitis and gastric ulcers, and also have hepatoprotective effects, for example, treating jaundice and cholestasis. Iridoid glycosides, which include geniposide and genipin, have remarkable hepatoprotective effects in three main areas: anti-inflammatory, antioxidant, and anti-apoptotic. Genipin has been shown to attenuate fulminant hepatic failure, and also genipin and geniposide together has been shown to have hepatoprotective effects against ischemic and perfusion injury in terms of decreasing oxidative stress and apoptosis. In terms of the pharmacokinetics, the peak plasma concentration of this geniposide would be achieved at 0.5 hours after per-oral administration, and half-life would be more prolonged with per-oral administration compared to IV form. And the tissue distribution, the peak plasma concentration would appear at 0.5 hours in liver and spleen, and 2 hours in kidney and brain. Geniposide toxicity could be divided into acute and chronic one. It is generally accepted that geniposide is a relatively safe substance to use since its LD50 is as high as 1431.1. And in terms of hepatotoxicity, it is shown that geniposide have to be as high as 574 milligrams per kilogram in order to have an increased change in ALT level. In terms of the chronic toxicity, a research from Shanghai University, China, found that there is no significant increased change in serum ALT, AST, ALP, and total bilirubin levels and relative liver weight. Therefore, it is concluded that geniposide at normal dose is unlikely to cause liver damage regarding to blood biochemistry and liver weight. So this slide would compare the normal liver with the hepatitis 1. As you can see on the left side, the normal liver would present with normal lobular architecture and cell structure, and there would be no evidence of centrilobular necrosis. While on the right side, hepatitis would present with atypical hepatocytes arranged in thick cords and inflammatory cells infiltration along with necrotic debris. Now let's take a look at the biomarkers. First one is the AST and ALT, which are the liver enzymes that act as a biomarker for hepatocellular injury. So when there is hepatocyte cell death, increased serum level of AST and ALT would be observed. Next marker is interleukin-6. IL-6 is a member of inflammatory cytokine that induce acute phase response. So Kupffer cells in liver could produce IL-6 too. So if there is increased IL-6, there is likely to be inflammation. Next marker is reduced glutathione, or short for GSH. GSH is one of the most important cellular antioxidants, which is most abundant in liver. It regulates oxidative stress, detoxification, and immune function. It also plays a role in approximately 10% of paracetamol metabolism. As a result, people with impaired level of glutathione, whether it's malnutrition or advanced cirrhosis, may be more susceptible to paracetamol toxicity. Next is hepatic MDA, or malondialdehyde. It is a marker of oxidative stress. Oxidative stress could occur from acetaminophen also. And in this study, hepatic MDA would be measured from fresh hepatic tissue. TNF-alpha is another inflammatory cytokine in which its level could be increased in inflammation. Now, this is the literature review slide. Let's look at the past studies about the geniposide and hepatoprotective applications. There has been one previous study that investigated the role of geniposide in protecting hepatocytes from acetaminophen hepatotoxicity by down-regulating CYP2E1 expression and inhibiting TLR4 in FKB signaling pathway. In this study, the design is that the geniposide is used as a pre-treatment before APAP or acetaminophen or paracetamol administration. The dose of geniposide used in this study is 10, 30, and 100 milligrams per kilograms in C57 mice. So the geniposide is administered 3 times, 8 hours apart each, and after the third dose is administered 8 hours later, the 350 milligrams per kilograms of APAP would be administered into the mice. 18 hours later, the mice would be sacrificed after being exposed to paracetamol. The results of geniposide pre-treatment from the previous study shows that there's a decline of markedly increased AST and ALT and also attenuation of pathological lesions in the liver. Also, there is a reduction in inflammatory cytokines level. In the previous study, the 100 milligrams per kilograms of geniposide is shown to have the most obvious effects. Other literature reviews shown that geniposide have many hepatoprotective effects in many diseases related to the liver, whether it be the non-alcoholic fatty liver, alcoholic steatosis, stellate cells activation, and CCL4-induced liver injury. Moreover, geniposide is shown to have protective effects against hepatic ischemia and could suppress hepatic glucose production and protect LPS-induced hepatic failure in mice. Next, let's take a look at our research question and hypothesis. The research question is, does Gardenia jasminoides fruit extract reduce hepatitis in mice with paracetamol overdose? And our hypothesis is that Gardenia jasminoides fruit extract can reduce hepatitis in mice with paracetamol overdose. For the objective, our primary objective is to investigate the role of Gardenia jasminoides fruit extract in attenuating paracetamol overdose-induced hepatitis. And secondary objectives is we would like to compare the effects of low and high dose of JG fruit extract on treating the paracetamol overdose-induced hepatitis. And we would like to identify the exact anti-inflammatory and antioxidant mechanisms that the fruit extract used to attenuate the liver injury in paracetamol overdose. Our methodology. The animal used in this study would be ICR mice, which is male, 5 weeks old, and weigh 30 grams. For the Gardenia jasminoides fruit extract preparation, firstly, we would grab the dried gardenia fruit and soak it in distilled water. After that, we would collect the filtrate and soak the crude again in 50% ethanol. After that, we would collect the filtrate from step 1 and 2 and mix together and make it undergo evaporation using an evaporator. After that, we would use HPLC method in analyzing the geniposide content in the extract, in which we get a yield of 22.67 plus minus 0.26% of the drug extract. The paracetamol preparation, a single dose of 600 milligrams per kilograms of paracetamol would be dissolved in distilled water and given to mice per oral via intragastric tube. Conceptual framework of our study. So we would have a model of paracetamol overdose mice, and this would cause a relative depletion in GSH and formation of NAPK hepatic protein adduct. Therefore, what happened is that there would be a reduction of natural antioxidant or decreased level of GSH, and inflammation, oxidative stress, and hepatocyte apoptosis would be reflected through increased level of TNF-alpha, MDA, IL-6, AST, and ALT. These two would contribute to hepatitis. However, with the presence of Gardenia jasminoides fruit extract, we would inhibit the reduction of natural antioxidant along with inhibition of inflammation and apoptosis. Therefore, we believe that Gardenia jasminoides fruit extract could improve the hepatitis. For the experimental design, we would use a total of 24 mice in our study, and we would divide them into 4 main groups. For the group 1 is control, we would feed them with distilled water, and at the end of the experiment, we would sacrifice them. For group 2 is an APAP group. We would feed them with 600 milligrams per kilogram single dose of paracetamol at the beginning of the experiment, and after 24 hours, we would sacrifice them. And for group 3 is APAP with low-dose JG extract. Low-dose JG extract, we would use 100 milligrams per kilogram geniposide for 2 doses. And group 4 is APAP with high-dose JG extract. For high-dose JG extract, we would use 200 milligrams per kilograms of geniposide for 2 doses. And the timeline for group 3 and 4 would be demonstrated as follows. So after administered APAP, 30 minutes later, we would administer first dose of geniposide. And 8 hours after the first dose, we would give the mice second dose of geniposide. And after 15 hours, 30 minutes after second dose of geniposide, we would sacrifice all mice. And it is also noted that the yield of geniposide from extract is 22.67 plus minus 0.26%. In terms of the experimental protocol, after we had successfully sacrificed the mice, we would collect the liver and the serum from the mice. For the liver, we would divide them into 2 parts. The first part would be freshly stored at minus 80 degrees Celsius to further use for hepatic MDA analysis and hepatic GSH analysis. And liver, another part, would be put into formalin and paraffin block for tissue pathology. And the serum, we would divide them into 2 parts. First part is for ELISA. We would run ELISA for IL-6 and TNF-alpha. And also another portion of serum would be used to run AST and ALT assays. For the histopathology grading in the APAP-induced liver injury, there are a total of 4 grades from 0 to 3. Grade 0 is the normal histology. Grade 1, minimal congestion and necrosis of single hepatocytes would be seen, but it is limited to the area immediately around the centrilobular vein, as the arrow points. For the grade 2, there would be moderate congestion and hemorrhage of the area around the centrilobular vein and extending into the mid-zonal cells. Most lobules would be affected. Areas of the confluent necrosis, however, would still be limited to the liver cells surrounding the centrilobular vein. In the grade 3, widespread areas of congestion and hemorrhage in the centrilobular and mid-zonal areas of the liver would be found. Confluent coagulative necrosis involving all hepatocytes in the centrilobular zone. Bridging of areas of necrosis between centrilobular zones would be common. And the arrows demonstrated that. So the expected benefits and applications would be to understand the effects that JG fruit extract has on treatment of paracetamol overdose-induced hepatitis. Moreover, we see this as a potential use of the fruit extract as a novel therapeutic agent in the disease. And also we would like to understand the underlying pathophysiological mechanism so as to use it as a principle for future drug development. So this is a brief time schedule for my project. I would like to finish it in 12 months in total. And for the take-home messages, I would like to ask all the listeners, through which mechanism does Gardenia jasmine or this fruit extract is hypothesized to exert its protective effect against paracetamol-induced hepatotoxicity? One or more answers might be correct. Now we have come to the end of the presentation. Thank you very much for your attention. Thank you.